Hi everyone. How are you all? Good evening. Can everybody hear me clearly now today? All right, Palak has joined, Lavanya has joined. So where did we stop? Where did we stop? Last class, I think there was some issues related to voice and all. That's not there. Okay, doubts. So I'm going to start with the male reproductive system again, because that time, there was some disturbances in between and we couldn't complete. Okay, so today we will start with the male reproductive system. And we'll see the slides on the diagrams of the male reproductive system. So everything was clear, like sexual, asexual reproduction, introduction and everything. And as I told you that the main parts of the male reproductive system are the main parts of the male reproductive system are the primary sex organs. Okay, the primary sex organs, the ducts, that means the accessory ducts, the accessory glands, and the external genitalia. All right, so these were the four parts I had told you, and we had stopped right here. Now we are going to continue from here. Okay, and Palak, you had some doubts, so before we start, just clear that out. Palak, just tell me what your doubt was so that quickly we can just start the class for today. Shall we start now? Okay, so this slide, as you can see, shows the male reproductive system consists of paired testis, accessory glands, and a duct system and penis. So what I told you was, the same thing is given over here. So you have this comes in the external genitalia. So you have the accessory glands, you have paired testis, which is the primary sex organ. Then you have accessory glands. There are three accessory glands, I'll tell you. There is this prostrate, then there is seminal vesicle, then there are Cowper's glands. You can see, So this is a sectional view, one side view of the male reproductive system. And they have just shown like the sectional view, one side view. So this is one testis. And you can see that there are these ducts coming from the testis. This is like coil duct on the head part of the testis, that is epididymis that they've told. And this is also, they've labeled, sorry. And this is also an important duct system. So this epididymis and this tube, this comes is the vas deferens. This is the main sperm duct. This is only cut and ligated during vasectomy. All right. So this duct comes all over the urinary bladder. This thing is the urinary bladder, and just focus on the tube that comes and just coils, and you can see it comes and meets. It's just look at the pointer over here. It comes and meets. There is a gland over here, seminal vesicle. So accessory gland is the seminal vesicle. So there's a duct of the seminal vesicle. This vas deferens meets the duct of the seminal vesicle. Okay, it meets the duct of the seminal vesicle, and over here you can see that it just goes and then joins the urethra. It goes and joins the urethra. All right. So over here is the meeting point of the this is the vas deferens and the duct of the seminal vesicle, and then these two join and form the ejaculatory duct. All right. So again, I want to explain all this, and then they join the main duct. I mean, they join this urethra, and the urethra, in case of male, the urethra is the common passage for urine and sperms. So it just comes and joins and then goes into the penis, which is the external genitalia, all right, glans penis. So this is roughly what I was telling you. The paired testis, which is the primary sex organ, because it produces the gametes. Then there is accessory gland. Now here you can already see you have this seminal vesicle, that is one gland. You have prostrate. Now prostrate is unique, I'll tell you. It is like only one in number. whereas seminal vesicle are 2 in number, but over here only one side view is given, so you are unable to see. Prostrate is there. Then you have Cowper's gland also, which is not shown over here. So in the coming up diagrams, I will tell you. And then you have the duct system, so you can easily see that we have this epididymis, which is the duct system, one of the duct systems. Then you have the main duct, which is vas deferens. All right. So this is the duct system. Apart from that, if we take a cross cross section or TS of the testis, it is further divided into many ducts that I will take up in the coming up slides. So a duct system and a penis, which is the external genitalia. So I hope this is just making you familiar with the parts of the male reproductive system. So this slide is clear to you. I hope so. Is this slide clear to you? Okay, let me just see. What is, I have a doubt. What have you written? What spelling have you written? Just write the spelling properly. What have you written? Tenderness. Lavanya says it's clear. Pramod, Ria. Okay. Palak, I cannot get you. What spelling have you written? I mean, exactly what do you mean? Next is, next slide is your, yeah, again, a better clear diagram. A better clear diagram we could explain over here, and you can just see the testis. This is the testis. This is the testis, which is the primary sex organ, and testis is present in a muscular pouch which is called the scrotum. Now I told you why the testis is present in the scrotum. Because see, scrotum is a muscular pouch-like structure. It's hanging outside. So testis is extra abdominal in position. We know that testis is the position of the testis. What is the position of the testis? It's extra abdominal. All right. So what do you mean by extra abdominal? It's not inside the abdomen. It's not intra-abdominal. It's extra abdominal. Why so? Why do we write so that it's extra abdominal? Why do we write that the testis is extra abdominal? The testis is extra abdominal because sperm production storage requires a temperature 2 to 3 degrees Celsius less than the body temperature. 2 to 3 degrees Celsius less than the body temperature. Okay, so why do we require um, scrotum? Scrotum is required because sperm production storage and maintenance requires a temperature 1 to 3 degrees Celsius less than the body temperature. Okay, that is why sperm production is very sensitive, and hence we say that testis is extra abdominal in position. So what doubts you have? Let me just see. So are the testes present? Are the testes present? Outside the abdomen. From the day one. I mean, when the male fetus is there, from that time, or it is like later later during the this thing they become. I mean, what is it exactly that happens? What is it that exactly happens? Okay. So in this slide, you can see that testis produces spermatozoa and secretes the male hormone. So what I want to explain you is testis is extra abdominal in position. It is extra abdominal in position. So you can see because sperm production requires a temperature 2 to 4 degree, or it's like some books write 2 to 3 degrees Celsius lower than that of the abdominal cavity. Otherwise, the sperms will be destroyed. Now see what happens. Testis is formed during the 7th to the 8th week of pregnancy. So when is the testis formed? 7th to 8th week. So when the embryo is 7 to 8 week old, that means that time only inside, if that is a male fetus, then that time scrotum. So why is it so, and how do they descend? How do they descend? Okay. So what happens is there is a passage called the inguinal canal. There is a passage called the inguinal canal, and through that passage, the testis descend into the scrotal sac. And gubernaculum is again, it's a ligamentous connective cord. It's a cord, it's a connection that connects the testis to the scrotal sacs. So last class I had told how the testis, I had explained you about inguinal hernia, all right. So over there you saw that there were spermatic cords, and spermatic cords were literally helping the testis to hang inside the scrotum, all right. And then there was this passage called the inguinal canal, and you had the partition. You had the partition, and then the spermatic cord was here, and the testes were like hanging by the help of the spermatic cord. So spermatic cord helps in placement of the testis into the scrotum. And another thing is there is gubernaculum. This is the ligamentous connective cord that connects the testis to the scrotal sac. Okay, so gubernaculum and spermatic cord, you have to remember. So these are the structures by which the testis is placed inside the scrotal sac. Okay, and we also discussed about failure of the testis to descend into the scrotum is called cryptorchidism. Cryptorchidism is a disease where the testis fail to descend into the scrotum. So what happens in such okay, so there's a question over here. This is a NEET question, that such a person, such a male in which the testis fail to descend into the scrotal sac, will they produce testosterone or not? I mean, what will be the possible abnormalities which that person will have? So will sperm production take place? Okay, will sperm production take place? Will testosterone be produced? Testosterone is the male hormone, all right. So will testosterone be produced? So they were just asking these options in the question. And now just write in the chat box and tell me, sperm production will be affected, or testosterone, will that person be producing testosterone or not? My question is that. Okay, so what happens if cryptorchidism takes place and testosterone is produced or not, is my question. Spermatogenesis, which was yes, FSH is responsible for spermatogenesis. FSH, I will tell you the hormonal control of spermatogenesis. There is FSH and LH. LH name is LH's name is different in case of male. It's ICSH. It's interstitial cell stimulating hormone. I'm going to tell you yes, LH as well as FSH. So LH and FSH, they are required for spermatogenesis. I will tell you about the the hormonal control of spermatogenesis in the coming up slides. Okay, so yes. So you are right, Pramod, you are right. Sperm production will be affected because sperm temperature is not proper for the development. But testosterone will be produced because testosterone production requires, what does it require? Testosterone, where is the testosterone produced and who produces the testosterone? The testosterone is produced by interstitial cells.
Only testis.
Yes, present. So testosterone is going to be produced, but sperm production will be affected because the temperature is not going to be suitable, because sperms are sensitive to temperature, and your body temperature is high, so sperms will be affected. And that's why when we discuss about low sperm count reasons, so you will see that all those people who are working in a very you know, where the temperature is very high, or they are like most of the time they are exposed to blast furnace or all these things, in them the sperm count is low, and there are other reasons also. Okay, so testosterone production will take place. All right. Okay, so next is oritectomy. I told you this term, you're familiar with this term. It is the surgical removal of the testis, also popularly known as castration. You know that ox and bull, I told you. Okay, so this aggressiveness is going to go if testis is removed, because the male hormone is also responsible for that aggressive nature. All right, and then they have described the structure of the transverse section of the testis, which I'm going to explain you properly. So just in this slide, what you have to understand is about descent of the scrotum during the 7 to 8 month of the pregnancy. Okay, so just I'm asking you a question, you tell me the answer. During which month of the pregnancy does the testis descend into the scrotal sac? Come on, write your answer in the chat box and send me fast. I am asking you, my question is that during which month of pregnancy do the testes descend into the scrotal sac? No, I just told you 7 to 8 month. Yes, very good, Ishita. Correct, Ishita. 7 to 8 month. 7 to 8 month is the answer. Okay, so now you followed, Ishita. You are right, and Arpit, it's 7 to 8 months. Just correct your this thing where you've written. Okay, so till here it's all clear to you. Till here it's all clear, everybody. All right. Okay. Thank you so much. Thank you so much. Okay. So then these terms are clear to you now. About the ts of the testis, so very carefully listen to me. If you take a transverse section of the testis, now till now we saw the side view, and I'm going to show you slides of the front view also. So if you see take a ts of the testis, what will you see? The ts of the testis is going to reveal a very important structure, which is the structural and functional unit of the male reproductive system, and it is the seminiferous tubule. It is the seminiferous tubule. Okay, so the ts of the testis is going to show you the seminiferous tubule, and how and what is there inside the seminiferous tubule we are going to discuss. So when you take a ts of the testis, you will find the testis. If this is the testis, you will find these small circles are seminiferous tubules, and what is there inside the seminiferous tubule we are going to discuss in the coming up slides. And each testis has approximately 750 seminiferous tubules. I told you, how did we get this number? Last class I had explained you, and you are going to explain this, that how did we get 750. I am going to wait for your answer. You're going to write on the chat box and send me, because each testis has 750 seminiferous tubules. All right. And what is there in the seminiferous tubule? Each seminiferous tubule has two kinds of cells. You have Sertoli cells and you have the germ cells, which are spermatogonia. Sertoli cells are also called sustentacular cells. They sustain the the sperms. That's why they are called sustentacular cells. And in between them, like outside the seminiferous tubule, you have these small short dots that I'm making. These are cells of the Leydig. So this is just a rough structure. So these are the structures that produce, the cells of the Leydig are the structure that produce the male hormone. The male hormone is not produced by the seminiferous tubule. So never write this MCQ wrong. Inside the seminiferous tubule, the sperms are formed. So you will find various stages of the development of sperm inside the seminiferous tubule. while between the seminiferous tubules you will find interstitial cells. Alternative name is cells of Leydig, and this is also called interstitial cell. All right, so this is also called interstitial cell. So what is the what is the function of these cells of Leydig or interstitial cell? Its function is to produce male hormone testosterone. All right, and then all this the network and all the alternative names and what path the sperms take, I'm going to explain you. So till here, is it clear? All the terms of this slide are clear to everybody? Yes, Lavanya says yes. Pramod says yes. All doubts clear till here? Shall we proceed now? Okay. Nice. Now, next we are going to study. Next, we are going to study the TS of the testis. You can just look at the words which are given in the slides. Just a second. All right. So before we just look into all this text, and you will don't get scared of all this. This is like very easy. Yes, just look at this diagram. Just look at this diagram. This is actually what I was explaining you, or even a clear diagram. So I'm going to explain this, and then there's a clearer diagram which is I think yes, this will be quite enough to explain you. So look at this diagram. This is inside of the testis. Okay, so this is what is there inside the testis. So can you see this tunica albuginea? The covering of the testis is tunica albuginea. This is very important. Tunica albuginea is the connective tissue, is the tissue covering the testis, and at places the tunica albuginea moves into the testis and divides the testis into many lobules. So just look at this. What am I doing in the red color? Can you see these lobules? Okay, so you can see that the tunica albuginea at places has entered the testis and has divided the testis into lobules. Okay, so there are how many lobules inside each testis? There are 200 to 250, or sometimes 300 is also written. 250 lobules inside the testis. Okay, so how were these lobules made? They were made by the covering tunica albuginea. All right, so 200 to 250 lobules, and each lobule, now just focus. I'm going to take a different color to explain you. So just focus on what is there inside the I'm going to take this dark color or this green color. All right, so just look at these structures, the green colored ones. These are tubes which are convoluted. Convoluted means which is like not straight, muda hua, bent. देखो, inside the red wala is the lobule, और उसके अंदर ये जो मुड़े हुए structures हैं, these are the seminiferous tubules, which are the structural and functional unit of the male reproductive system. So each male reproductive system has 750 seminiferous tubules. How come? Because 1 to 3 seminiferous tubules, I'm just writing SMT, you understand, seminiferous tubules, present in each lobule. Okay, so all this you have to be very particular about, because you will lose marks if you don't do it properly. So 1 to 3 each lobule. Now there are 250. Now just take the maximum number. There are 250 lobules, and each lobule has got 1 to 3. That means the upper value is 3. So 3 seminiferous tubules are present in each lobule. So then total number of seminiferous tubules in each testis is going to be 750. So I asked this, but I myself I have answered this. Okay, there will be questions for you, don't worry. So till here, have you understood that, you have the testicular lobules and then you have the seminiferous tubules, all right? Now you can see that from the seminiferous tubules, you have um, another structure coming up, which is like a net kind of a reticulum kind of a structure. This is called retitestis. This is called retitestis. So the sperm has been formed inside the seminiferous tubule. Then it comes to the retitestis, which are a network of tubes. And then from the retitestis, can you just see these very thin tubes. These very thin tubes are called vasa efferentia, the ones in blue that I'm making, vasa efferentia. All right, so this is vasa efferentia. So from the retitestis arise the vasa efferentia. These are again fine thin tubes. And and finally, the vasa efferentia forms another tube which is highly convoluted, and this is the orange one is epididymis. The epididymis has three parts: the head part, caput, the body, and the tail, chorda. So this is the epididymis, and after the epididymis, you can see this tube. This is your vas deferens. All right. So this is like it looks very complicated, but if you do it properly, it's not like that. So let me just write the path which the sperm takes. So once more with me. The sperm production takes place in the seminiferous tubule, then the sperms travel to the retitestis, then they travel to the vasa efferentia. Vasa efferentia is different, vas deferens is different. Vasa efferentia, then into the head of the epididymis. Caput is the head, corpus or body is the second part, and chorda is the tail part. All right, and then it goes into the vas deferens, and from the vas deferens, it takes that path. It meets the seminal vesicle and all those parts. All right. So let us just write all the parts in order, or the path which the sperm takes. So you have these seminiferous tubules. Okay, so ST means seminiferous tubules. From the seminiferous tubules, it goes into the sperm goes into the retitestis. Retitestis is RT. RT is retitestis, and then it goes into the vasa efferentia. I'm going to write vasa efferentia because you will make mistake. Vasa efferentia. Okay, so vasa efferentia, and then you have. Vasa efferentia के बाद you have epididymis. EPIDI EPIDIDYMIS is epididymis, and from the epididymis it goes into the vas deferens. It goes into the vas deferens, right? And from vas deferens, it takes that full route. It goes into the, via penis, all the path it takes, and then external genitalia, say it comes out. So have you followed this? You are asked questions in the NEET to tell the pathway, external genitalia or the penis you can write. All right. So I hope this much is clear to you. Just write on the chat box whether you've understood this much or not. All clear. You can take time and understand. Clear, all clear. Oh, thank you so much. You're understanding everything. It makes me so happy, really. Palak has understood. All well. Is there anybody who has not understood the path? Cleared. Ria also says cleared. Very good. Okay. So till here it's all clear. It means, okay, Pramod, I'm just reading it. It means that all these structures do only transport of sperms. Okay. One thing I want to explain you. Yes, all these structures. Now, all these structures, they are, see, seminiferous tubule is making the sperms. Okay. We will read spermatogenesis in the coming up slides. Seminiferous tubules is seminiferous tubule is is the place where the sperms are produced, Then retitestis is just a small network of tubes where it travels. Vasa efferentia is again meant for traveling, you were right. Epididymis, yes. Primarily, the sperms are stored in the epididymis, and then they are secondarily stored in the vas deferens. So there are two places where the sperms are stored, in the epididymis also and in the vas deferens also. All right, but the sperms which are here, if you just take these sperms from here and make it fuse with an ovum, they will not fuse because they do not have that much of energy and motility. So all that is acquired when the sperm is mixed with the secretion of the accessory glands. Okay, now in the chat box, you just write the answer to this question. Where are sperms secondarily stored? There's only one answer which has come up. No, epididymis, it is only stored. Albuginia point. All right, I will explain it again. See, epididymis, it is stored, and then as it travels, it is also stored in the vas deferens. But see, I want to explain you something, that all the time, see, sperm production is a continuous process. So once the male attains puberty, sperm production starts. So sperm production is taking place continuously, but all the time the male is not ejaculating. So where do the sperms go? There are phagocytic cells in the epididymis which digest these sperms. So yes, this is the place. Epididymis is the place where the sperms are primarily stored. Like the first storage point is epididymis, and secondary point of storage is the vas deferens. Did you understand now? Yes, you're right. Tunica albuginea is the connective tissue membrane that covers the testis. Clear? Shall I just move to the next slide? Okay, all right. Tunica albuginea is clear to you? Who asked the doubt? I think Pramod asked the doubt. Is it clear to you, Pramod? Okay, good. So we will move to the next slide. So we have done till now we have done the structure of the testis, the side view I showed you. Then I told you about the covering of the testis, and then the TS of the testis. Ma'am, whole point, outer covering and inner. I didn't get you, Arpit. Whole point as in outer covering. Tunica albuginea is the covering, and that moves into the testis and divides the testis into lobules. Okay. Now it's clear. Now just look at this diagram. This is the diagram where you again see the TS. So you've seen the internal structure earlier, and now this is a magnified, a closer view of the seminiferous tubule. So this is one seminiferous tubule. All right. So this is seminiferous tubule. What do you have inside the seminiferous tubule? So this is testis, and this is seminiferous tubule. So inside the seminiferous tubule, now just look at very clearly. You have two kinds of cells. You have these cells, which are the Sertoli cells or sustentacular cells, and you have another cell which is like lining this seminiferous tubule, which is the germ cell, which is the spermatogonium. So what are the two cells present inside the seminiferous tubule? You have the Sertoli cell, which is like very important. I'll tell you the importance of Sertoli cells. So Sertoli cells are like very important, and they support, protect provide nourishment and activate the sperms. Activate as in, all the time during spermatogenesis, the sperms, after being produced, they are embedded into the membrane of the Sertoli cells. And this also, um, serves as a barrier, because the germ cells which are like here, you know, the germ cells do not come in direct contact with the blood. Otherwise, there will be, an immune reaction. So Sertoli cells are like, nurse cells. They provide all the support, all the nourishment, and they also take away waste products from the spermatogonium, okay, into the blood. So they also form a blood testis barrier. So you have the Sertoli cells, and you have the spermatogonium. Okay. So these two cells are clear to you. So here in between you have, this is the Sertoli cell, this is the Sertoli cell, right? And then you have, what do you have? This is your, which is your spermatogonium, the entire epithelium, the entire structure, all these cells, which you can see, these are the, this is these cells which are lining. So like, if you have to make, it will be like this. These cells which are like elongated, these are the Sertoli cells, and look at this epithelium kind of a thing. These are the germ cells, and they will, so this is the spermatogonium and this is the Sertoli cells. All right. Now, what is going to happen over here? The spermatogonium is going to undergo spermatogenesis to form these sperms, and the sperms will be nourished, supported, and protected by the Sertoli cells. All right. So how does that happen? We are going to discuss. Is my voice not clear to you? Is my voice not clear, not audible? Did you understand till here? Just focus on Sertoli cells and spermatogonium. Focus on Sertoli cells and spermatogonium. That much is clear to you? Then we will proceed. Is that much clear to you? We are just concerned about tunica albuginea. We are just concerned about tunica albuginea. Tunica albuginea is that connective tissue cover on the testis. Concentrate on that. Sertoli cells are the cells which are present inside the seminiferous tubule that support the various stages of sperm production. The name is Sertoli based on the person who had first seen it. I think it's mentioned in the slide. This Enrico Sertoli was an Italian histologist who had first seen the Sertoli cells. So the importance of Sertoli cells, I'll tell you. So just you understand the location over here. These are the Sertoli cells, and between the seminiferous tubules, like outside the seminiferous tubules, you have the interstitial cells. So interstitial cells are the cells that produce your male hormone, and this has nothing to do with the temperature. So even if the testes do not descend into the scrotum, testosterone production will continue, but yes, sperm production will be affected. I hope this much is clear to you. Is this much clear to you? Okay, let me see what you've written in the chat box. Yes. Okay. All right, then we'll move to the next slide. So what are the two kinds of cells present in the, what are the two kinds of cells? Present in the seminiferous tubules. You have the Sertoli cells and you have the, yes, Sertoli cells, and you have the spermatogonium. Now what happens to the spermatogonium And what does the spermatogonium exactly do? And how are the sperms produced? Just concentrate on this slide. So inside the seminiferous tubules, okay, let me just see what doubts you have. Second type of cell, which cell you are referring to? Very good, very good. Janisha, you've got it. So what are the second type of cell? Second type of cell is the spermatogonium. So Sertoli cell and spermatogonium. Okay. Okay, so shall I explain you spermatogenesis now? Everybody? Okay. Let us do spermatogenesis now. Now listen, there are certain terms which are very, very important when you study spermatogenesis. What is spermatogenesis? Spermatogenesis is the production of sperms. So what does spermatogenesis mean? Spermatogenesis means production of sperms. So I will just take a white screen to explain you. Okay, so let us just, and this is like very important. So I would request everybody to write in their notebooks as we are doing it. So spermatogenesis is what we are studying. Spermatogenesis. All right. So what is spermatogenesis? Spermatogenesis is the production of sperms. Spermatogenesis is the production of sperms. And what happens? You have the spermatogonium. So you can write over here, production of sperms. The entire process is referred to as spermatogenesis. Now, what are the steps? What are the steps? Now, in the testis you have immature germ cells called spermatogonia. I told you spermatogonia, all the entire lining they were present, no? So spermatogonia, what are spermatogonia? These are the germ cells. These are the germ cells. And what is their ploidy? They are diploid. They are diploid. So spermatogonia are the germ cells present in the seminiferous tubules. Now, these immature germ cells or the spermatogonia produce sperms by spermatogenesis that begins at puberty. Getting everyone? That begins at puberty. So first step, what happens? They are, and you know where it is present on the inner wall of the seminiferous tubules. So they undergo mitotic divisions. They undergo mitotic divisions. You know about mitotic divisions, right? Mitosis is a division, equational division that that helps in the production of cells where the chromosome number doesn't change. So from one cell you get two cells, but their chromosome number doesn't change. All right. So spermatogonium undergoes mitosis. So it undergoes mitosis to produce more number of spermatogonia. So this undergoes mitosis to produce, okay, it undergoes mitotic division and increases the number of spermatogonia. So you have more and more spermatogonia. So it's all right. You can just simply write mitosis and more number of spermatogonia produced. Now, at the same time, they undergo growth. So they increase their number, divide by mitosis, increase their number, undergo growth, and now they are called primary spermatocytes. So some of the spermatogonia are called primary spermatocytes, the ones which show growth. You understanding? Primary spermatocyte. So see, what did I tell you? Spermatogonia are the male immature germ cells. They are present on the inner wall of the seminiferous tubules. And we are talking about these cells. You have this seminiferous tubule and you have these spermatogonia. So we're talking about this. We are not talking about, this is the Sertoli cell. So nothing happens in the Sertoli cells. These just support the sperms. So these cells are spermatogonia. So they undergo mitosis, increase their numbers, and some of the spermatogonia, they show growth, and they are called primary spermatocytes. Now, what happens to the primary spermatocyte? The primary spermatocyte undergoes meiosis. Meiosis is a reductional division. Come on, you know this. So it undergoes meiosis, and as a result of meiosis, it in the meiosis one, you know that the chromosome number is half. So let us just discuss about the ploidy. Spermatogonia were diploid, so I've written 2n. Over here also the ploidy didn't change. Now this is the reductional division. Over here the ploidy is going to change. The ploidy is going to change. All right. So now what happens is that meiosis 1 results in the formation of secondary spermatocyte. All right. So secondary spermatocyte. Now this is meiosis 1, and we have read that meiosis 1 is a reductional division. So what is going to happen to the number of chromosomes? The number of chromosomes is going to be half. So the number of chromosomes was 2n over here, half is n. All right. Now secondary spermatocytes undergo meiosis 2. And after meiosis 2, what are they going to form? What are they going to form after meiosis 2? After meiosis 2, they are going to form spermatids. So after meiosis 2 takes place. So meiosis 1 has taken place. Now meiosis 2 is like mitosis. Over here, the chromosome number doesn't is constant. All right. So this is constant now. So meiosis 2 is like mitosis, doesn't change the chromosome numbers. This results in the formation of spermatids. All right. So what has happened till now? And over here, what will be the chromosome number? The chromosome number will be same. So you just ask the ploidies also. So let us just now do a quick recap. So we were discussing spermatogenesis. The production of sperms inside the seminiferous tubules at the time of puberty is called spermatogenesis. From inside the seminiferous tubules, you have immature germ cells that produce sperms, and these are called spermatogonia. So step 1 is, this is the step 1 where spermatogonia undergoes mitosis to produce their numbers, and multiple spermatogonia are produced. Some of these spermatogonia show growth, and they are called primary spermatocytes. Primary spermatocytes are diploid, and that is, they contain 46 chromosomes, and some of them the primary spermatocytes undergo meiosis 1, which is a reductional division, and they form secondary spermatocytes. And secondary spermatocytes further complete the meiosis 2, and now they are called spermatids. What happens after that? Now spermatids, they have not taken the form of a sperm. So the spermatids in the last step are transformed into spermatozoa. Spermatozoa are the real sperms by a process called spermiogenesis. Okay, let me just clear all the terms to you, because this is really asked in the MCQs. So let us just write once more properly and clearly. So we have spermatogonia. We have spermatogonium. Singular is spermatogonium and plural is spermatogonia. And this is primary spermatocyte. Primary spermatocyte forms secondary spermatocyte. Secondary spermatocyte forms spermatids. And spermatids mature to form spermatozoa. Please be very clear with these terms. Or sperms. Now let us just, the entire process, this whole process is called spermatogenesis. But yes, please concentrate that there are different names given to these processes. So the process of formation of spermatids, like spermatozoas, like from here to here, I'm going to answer your questions. From here to here, spermatids to spermatozoa is called spermiogenesis. Okay, so till here only this step is called spermiogenesis. And when after spermiogenesis, now I told you that the serotoli cells, the spermatozoas are embedded into the membrane of the serotoli cells. And when after spermiogenesis, when the sperm heads they become embedded into these sertoli cells, and when they are finally released from the seminiferous tubule into the lumen, this step is called the release of the the spermatozoa from the sertoli cell membrane into the lumen of the seminiferous tubule is called spermiation. Be very careful. You are very it's very natural to get confused between these terms. So this is called spermiation. The entire process is spermatogenesis. Spermatid to spermatozoa is spermiogenesis, and release of the spermatozoa. Release as in where? See, I am going to explain you. So just concentrate over here. You have this one you have one seminiferous tubules magnified, and this is your sertoli cells. Now over here, all these you know structures are being formed. So you have this spermatogonium, then primary spermatocyte, secondary spermatocyte, then you have spermatids, and then final uh, sperm is over here. So the head is like embedded over here. So the release of this into the lumen, release of the sperm into the lumen of the seminiferous tubule is called spermiation. Whereas transformation of the the spermatids into the spermatozoa is known as spermiogenesis. I hope these terms are clear to you, everybody. Now, what doubts do you have? Let me just see. What are your doubts? Just write down in the chat box and send me. Spermatid. Spermatid is a stage. Spermatid is a stage just before the spermatozoa. See, spermatid is like, when sperm has got a definite structure. It has got a head, it has got a middle piece, it's got a tail. So that appearance comes only during the spermatozoa stage. A stage before that, when it has not developed all that, is known as spermatid. Spermiation, you want me to repeat, Ria? See, spermiation is a process of release of the sperms or the spermatozoas from the membrane of the Sertoli cells into the lumen of the seminiferous tubule. I told you, because once the sperms are formed, their heads are embedded into the Sertoli cells, because they have to derive nutrition and everything from here. And once they are all fully formed, they are released into the lumen, and from the lumen they will be carried to the retetestis and all that pathway that I told you. Sorry. Clear now, Ria? Is it clear to you, Ria? Okay. So till here, it's all clear to you. So you will get questions. So this is also a neat question. All this spermiogenesis, spermiation, spermatogenesis, ploidy. Once again, let us just clear the ploidy once and for all. So this is going to be diploid. This is going to be diploid, because just this grows into this. This is meiosis 1. This is haploid. Again, this is meiosis 2. This is like same chromosome number, and you know that the sperms are haploid. So just be very clear. Be very clear with the ploidy levels, because they do ask you to match the ploidy levels. All right. So I hope spermatogenesis is clear to you, and I'll just show you the diagram. Just write on the chat box or not it is clear, so that I am assured that you've understood everything. Yes, clear, ma'am. And is there anybody who has any doubt related to the terms like spermiogenesis, spermatogenesis, spermiation? And if you get this in MCQs, you'll be able to do or not? right. So let us just quickly revise what all we have done in today's class, and then a quick just brush up of the topics. So what we did, I told you about location of the structure of the testis. We did structure of testis, path of sperm, the path which the sperm takes. So this was also clear. So all these MCQs you should be able to do. The structure of the testis, the path of the sperm. I told you about the duct systems, the duct system. All right. And then about the scrotum also we discussed. Then we discussed about spermatogenesis. We discussed about spermatogenesis. So just summing up, I will tell you that serotolin cells are very important cells. And you know what? The FSH, which is produced by the anterior pituitary. Remember, the gonadotropin releasing hormone. It produces two kinds of hormone, the FSH and the LH. So this FSH actually binds to these serotolin cells, to maintain a proper concentration of hormone that is required during spermatogenesis. So serotolin cells are very, very important, and this LH binds to the interstitial cells, and that's why it is called interstitial cells stimulating hormone. Until and unless this binds to the interstitial cell, testosterone is not going to be produced. Okay, so in the coming up classes, I will tell you about the hormonal control of spermatogenesis. So we will stop over here. Hope you've understood till here, and I'll see you on Monday. All right. Okay, so thank you so much for listening patiently. Stay tuned to us, and we will meet next class. Thank you so much.